Hello, everyone. My name is Chris Griffiths. I have the privilege of being the president of the ESDR at present, and I'm a dermatologist in Manchester. And welcome to the second of our ESDR live events that we've called ESDR Kitchen. The first one was 2 weeks ago, and hopefully you were able to tune into the recipe book where you heard about research techniques made simple. We've got other variations on this theme. Today you're going to hear a debate between two protagonists in our Sweet and Sour series, and then I'll come on at the end of this today's seminar to discuss what you're going to hear over the next month. So I'm now going to hand over to my colleague on the ESDR board, Professor Ellie Sprecher, who's going to be Master of Ceremonies for today's Sweet and Sour debate. Ellie, over to you.
Yes, so good morning, good afternoon, maybe good evening, everybody, and welcome to this Sweet and Sour lecture on the role of sunlight in melanoma pathogenesis. So it must be 9:30 p.m. in Brisbane, Australia, from where David Whiteman will try to convince us that melanoma is all about the sun. David is a worldwide renowned medical epidemiologist. He has a special interest for the causes and control and prevention of cancer. He has received his medical degree from the University of Queensland in '91 and his PhD in cancer epidemiology in '97. He was awarded a Neufeld Medical Research Fellowship to undertake a postdoc training at the University of Oxford in cancer epidemiology. He returned to Brisbane in 2000 and he now leads a large program on cancer research comprising national and international studies of melanoma and other cancers. On the other hand, it is 12:30 in London, from where Julia Newton-Bishop will try to convince us that there is more than just the sun in melanoma. So Julia is a dermatologist who worked for many, many years in the Leeds specialist melanoma multidisciplinary team. This is a team which manages poor prognosis melanoma for the Yorkshire region of the UK. She's also a clinician scientist and professor of dermatology at the University of Leeds. She leads the melanoma research group within the Institute of Cancer Research at St. James, and her research group uses genetics in order to understand the susceptibility to melanoma and survival from melanoma. She's a fellow of the Academy of Medical Sciences in the UK, and she was awarded the Lifetime Achievement Award for Melanoma Research in 2017 by the Society for Melanoma Research. And now, without any further ado, David, the floor is yours.
Thank you very much, Ellie. I'm just going to share my screen here. And hopefully you can all see a slideshow there with my title, Melanoma, It's All Sun. So I'm speaking for the affirmative. So thank you for the invitation to present to the ESDR. It's a great privilege. I'm delighted to be beaming to you from Brisbane. But as we start this discussion of why melanocytes turn into malignant melanomas, I'm going to concede points to the negative side already and acknowledge that certainly host factors are very important in the etiology of melanoma. And we know that a range of phenotypic factors increase a person's risk of melanoma and also a person's genetic architecture. And I'm sure Julia will speak to that. So I'm here to convince you as to the importance of sun exposure and sunlight and how it affects melanocytes to become malignant tumours. And I'm going to quickly, in my 8 or 9 minutes remaining, just summarise the epidemiological evidence, the molecular, experimental and the randomised trial evidence. So when we think of the earliest epidemiological evidence that melanomas were caused by sunlight or this was a risk factor. I mean, the earliest evidence came actually from simple analysis of melanoma mortality in different countries around the world. This was done in the 1950s by Herbert Lancaster. And this observation stands to this day. And this just compares incidence rates in continents around the world. This is in the International Agency for Research on Cancer. And you can see that in the sun-drenched Antipodes, where the Australian and New Zealand population, predominantly European heritage, has melanoma incidence rates that far exceed those of comparable populations in North America and Europe. And when you drill down into country-specific incidence rates, we've got the countries of Europe in these top 4 panels, Australia and New Zealand in the bottom left, and the United States and Canada. you can see firstly that the antipody and incidence rates are markedly higher, but you can see also that in all of those countries over time, over the last 30 years, plotted on the same axis, there's been a steady increase in the incidence of invasive melanoma in all of those populations. And in cancer epidemiology, when you see changes in incidence and particularly rises in incidence, that's very strong evidence that there are environmental factors driving the incidence of cancer. Now, some people will say, quite rightly, that part of this is increased detection of melanoma, and that is true. There is undoubtedly some increased detection of tumours. But then if you look at the mortality rates, the age standardised mortality rates, both in European countries, Australia and New Zealand, and also in the US, there have been significant, though smaller, year-on-year increases in mortality from melanoma, even though we've had earlier detection and better treatment. So fairly convincing descriptive evidence that these melanoma incidence and mortality rates are changing over time in different populations. There's been a whole lot of other epidemiological evidence, and time is too short to summarise it all. But we do know the role of sunlight differs for different people. Some people require more sunlight than others to drive melanoma. And some people particularly require melanomas to be initiated early in life, with sun exposure in childhood being particularly problematic and a driver of melanoma. And as I mentioned, there's an emerging epidemiological concept that's been backed by molecular and genetic data that there are indeed multiple pathways through which sunlight causes melanoma. Some people need more, some people need less, but virtually all cutaneous melanomas have some degree of sun exposure as their cause. And the evidence for that really comes from more recent molecular evidence. So we've known for decades that ultraviolet B radiation induces particular characteristic photolesions in the DNA double helix, particularly at these dipyrimidine sites, leading to cyclobutane pyrimidine dias and these photoproducts. And if these aren't repaired by polymerases before cell replication, then you get fixed mutations. And the mutation burden of melanoma has been shown. This is quite an old slide now, but it documents a trend that's emerged, that if you look at all the different kinds of cancers and you plot their mutation burden here on the left-hand axis, this is on a logarithmic scale, but it shows you the amount of DNA damage per megabase of DNA in tumour samples. Melanoma is right up here at the extreme edge with a very high mutation burden. And I guess the really important thing is that compared with all other kinds of cancer, when you look down at this bottom panel, you can see that melanomas carry this very, very high proportion of C to T transitions, which are characteristic UVB signatures. Now, of course, some of these DNA mutations are simply incidental passenger mutations, but a high proportion of these UV-specific mutations occur in the driver genes that are actually causal for melanoma. Not all of them are UVB specific, that's certainly the case, but a very high proportion of them are. There's also experimental evidence, moving on from molecular to the more experimental. The best animal models we have are various kinds of mouse models. As an epidemiologist, I'll be the last person to argue that mice are the best models for human cancer, but they certainly support the notion that UV exposure is important at initiating these cancers. And particularly, they sort of recapitulate the idea that early life sun exposure is important. And so there's a body of evidence around that. And in human studies, of course, we can't induce cancer in humans through UV, but we can certainly induce melanocyte proliferation. And this is work we've done with Elke Hacker, where we irradiate human volunteers and look to see what the response is of their melanocytes. So when you have a control site, here you have people exposed to 2.5 MEDs of UV, solar UV, and then you have a site where sunscreen is applied first. If you take biopsies from those sites, this is the control site. You can look at the melanocyte density after 14 days. It's around 11 or 12 per unit area. 14 days after that UV exposure, you get this proliferation of melanocytes, about a doubling of the number of melanocytes in the basal epidermis. And that effect is completely abrogated by applying sunscreen first. So pretty good evidence here that when you expose human skin to melanocytes, expose human skin to UV, the melanocytes are induced to have a proliferative response. And that, coupled with mutations that can occur, is the sort of seed, I guess, for melanomas to arise. And then the last bit of evidence that I'll give you is that we know from the Nambour skin cancer trial that people who are supplied with sunscreen, applying it daily and reducing their amount of exposure to the sun have a systematically lower incidence of melanoma when you follow them up over a long period. This is a 15-year time course. The top line shows the control arm, people who just had usual care, and their cumulative incidence of melanoma. The bottom blue line is showing the incidence of melanoma in the arm randomized to receive daily sunscreen. So that trial wasn't powered for melanoma as an endpoint, but it's the only randomized trial we have, and it shows that sunscreen appears to prevent the incidence of melanoma. So why such a focus on sunlight? Well, I hope I've convinced you that it explains the variation and incidence across both populations, across time, and I haven't gone into age, sex, and anatomical site, but I would like to convince you later that that's the case. And the magnitude of effective sunlight is large. You can see that across different populations, there's at least a doubling of incidence with high levels of sun exposure. And I guess the key factor for me as a public health physician is that it's the only modifiable factor that we have currently. We can actually modify people's exposure to the sun. And we can do that particularly by advocating the use of sunscreen. And I won't go through all of this, but there is good evidence that sunscreen prevents DNA damage, it reduces proliferation of cells, it inhibits the development of nevi, and it reduces the incidence of melanoma. So what we've been doing in our country, where melanoma is a particular problem, is trying to estimate and model how much benefit would we get if we could get the population to use sunscreen. So this dark blue line is the trend line for melanoma incidence over the next 15 or so years, with a steadily rising incidence of melanoma. But we would see if everyone used sunscreen as they should, then we might get a 33% reduction. That's an unachievable target. So what we'll probably be left with is a more reduced model where we get people to use sunscreen at an increasing level of 5% per year. And over a 15-year period, we might reduce melanomas by about 1/6. So I'll close there. I just want to leave with you that melanoma is a heterogeneous cancer, but a large part of the causality is due to sun exposure. And that evidence comes from both observational and experimental studies. And of course, it's a modifiable exposure. So we can actually control how much exposure people get. So from Brisbane, I will say farewell, and I'll hand over to my adversary, Julia.
Julia, please. You will show us why there is more than just the sun.
I will. There we are, finally. So I agree, there is no doubt that sun exposure causes most subtypes of melanoma, albeit not all. But here I'm going to talk about the variation in susceptibility to melanoma, which modifies our public health advice, or should do, and that the type of causal sun exposure actually remains a puzzle. So all dermatologists know what is shown in this slide from our case control study, that melanoma is more common in people with red hair, with type 1, type 2 skin and freckling, that it's associated with a family history sometimes, and that it's more common in people with lots of nevi. And these are clinically recognizable phenotypes that we're all familiar with. And in the Genomewide Genetics Consortium, we've done a lot of work over many years looking for those causal genes. And this is a genome-wide association study Manhattan plot. And these are the skyscrapers. These are the genes that are associated with melanoma risk. This one on chromosome 16 is actually truncated because that's much more strong. This is MC1R, the gene for red hair and freckles. And what we've done here is to color the peaks according to the phenotype. So all those red peaks are genes, inherited genes associated with pigmentation phenotypes. And there are a few, not so many as expected, in black that are associated with lots of moles, and the green ones are associated with a biological phenotype. So these are all genes associated with melanoma risk, which also predict telomere length. And telomeres are crucial bits at the end of chromosomes, here coloured in pink. And they perform crucial functions during cell division. They stop chromosomes getting mixed up effectively, and these genes also code for the length of telomeres. So this shows that pigmentation phenotypes, nevus phenotypes, and telomere function are all biological and clinical phenotypes associated with risk. So we understand a lot about that risk, but we don't generally show much success in conveying that to the population. And this figure here is an attempt to explain what I mean. So of course the sun is not an entirely harmful thing. It's a beneficial thing in many ways. It makes us feel good. It's crucial for the synthesis of vitamin D, and it has anti-inflammatory effects. So here what we're trying to do is talk about susceptibility in respect to this risk-benefit ratio associated with the sun. So what we've done is we've taken these SNPs, these genes we've identified to be associated with risk, and this diagram illustrates what might be associated with risk according to how many of these genes that you have. So in red, this is the population risk for the United Kingdom. So basically health prevention advice is based upon this level of risk. And what this diagram shows is that actually a lot of people in our population have very low risk in comparison with those at the bottom end here. So advice that we give generally actually is of limited value for those people, and indeed these people at the upper end of risk are perhaps not advised quite clearly enough of their relative excess risk. And essentially I think we should take this into account when we're giving public health advice. We have to try and convey something of the level of the risk, bearing in mind the beneficial effects of sunburn. Sunburn is crucial, and actually I've missed a slide here. This is from a case control study we reported in 2009, or rather a pooled data analysis of a clinical case of case control studies. So we took lots of data, kindly given by the authors from Europe, that's at the top here. We took data from middle latitudes, or studies performed in middle latitudes, and studies at low latitudes, very hot countries. And what we can see is that in this forest plot, we've tried to orientate the results according to melanoma on the trunk, melanoma on the limbs, and melanoma on the head and neck. And what we can see, these diamonds here, is that there is a clear relationship between sunburn and risk at all latitudes and at all body sites. So there's no doubt about that. And we saw something very similar for sunbathing, although it wasn't quite as clear. And this relationship between sunburn and risk was understood many years ago, and Fears et al. In 1977 put forward this intermittent exposure hypothesis, that it is intermittent exposure, sun exposure on holiday, that causes melanoma. But when we were doing these analyses, expected also to see an effect of cumulative sun exposure, but actually we didn't see any evidence when we did similar analyses for either occupational sun exposure or a measure of total sun exposure through life, and that is odd. We subsequently reported our own case control study, and in this we confirmed an increased risk associated with reported sunburn. We didn't see anything in relation to reported holiday exposure, and what we saw to our surprise was a reduced risk associated with weekend sun exposure. So remember, this is United Kingdom, which isn't the sunniest place in the world, but individuals who were reporting quite a lot of sun exposure at the weekends, or at least being outside at least 4 or 5 hours, Saturdays and Sundays in the summer months, had a reduced risk of melanoma, and there was something of a good trend there, and that was interesting. Subsequently, there was this study. This is a cohort study from the US, where they used where nurses lived in the cohort to predict how much day-to-day sun exposure they had. And this is the relative risk for basal cell carcinomas of about 2.4 for basal cell carcinomas. For squamous cell carcinomas, it was 2.53, but for melanoma, it was actually reduced, 0.68. So that's two pieces of evidence that some moderate sun exposure might actually be protective. And this is a big study performed by the GEM investigators, and they showed something similar. So they showed that occupational sun exposure was actually protective for melanoma on the head, as was a measure of total exposure on weekdays for melanoma over the upper limbs. So this is odd, especially when you take into account the fact, as reported earlier by David, that of all the cancers you see more mutations in melanoma, and they are generally speaking C to T, indicative of sun exposure. Now, these high mutation rate is especially true of melanomas arising on the head and neck and in the elderly. And in the two-root hypothesis that David's group has argued, the argument is that there are two routes to melanoma, that there are BRAF mediated melanomas, mainly in MOLI people having intermittent sun exposure, and that other melanomas driven by other driver mutations are associated with more sun damage in the skin. And there's no doubt that that is true. So how do we explain this? Firstly, we may see a lack of association with occupational sun exposure because sun-sensitive people might choose to work indoors. But it's also possible that photoadaptation may modify risk, is the argument I put to you. That there is a lot of data, particularly from animal studies, suggesting that when we get burnt, profound changes occur in the skin, which are not just mutational, but related to local immunosuppression. And moreover, in mouse studies, you can see that there is systemic immune suppression. And so an hypothesis is that photoadaptation may limit these profound biological changes whilst failing to prevent mutations. So it's an interesting puzzle. So we have that hypothesis, and I put to you this other hypothesis, and that is that we have become maladapted in some ways in the north to sunny holidays. So as man moved north from the equator, then we developed paler skins. And there are two arguments conflicting arguments about how this may have arisen. Firstly, that there was a positive selection for some variants in pigmentation gene, SLC45A2, which meant that in the north, man was able to manufacture sufficient vitamin D, but that photoadaptation evolved to avoid damage as a result of sun exposure. That was the balance there. The other population genetics argument is that as man moved away from the equator, then we lost the negative selection pressure for MC1R variants. But either way, we evolved to be paler, but yet we had a measure of photoadaptation, which actually sunny holidays on the beach is circumvented. We don't have time on the beach to develop that photoadaptation, suffer profound sunburn as a result, and melanoma may evolve. And so in summary, from this 10-minute whiz through this evidence, I would say I agree that melanoma is related to the sun, but the sun has beneficial effects, and we have to balance the harm and benefit, and we should target our prevention, if we can, to those who are particularly at risk. So it's easy to say when you go on holiday, don't get burnt. What's more difficult is the other things. We've seen, or I have mentioned, that solar damage is associated with melanomas, especially on the head and neck. However, the lack of epidemiological evidence for cumulative sun exposure and risk suggests that these tumours are arising in people who are very susceptible to the ill effects of sun exposure and don't perceive themselves to have had a lot of exposure in the past. Maybe they're having microburns through life. And the complicating factor is these data which I've reported, which suggest that photoadaptation may actually reduce melanoma in a proportion of people. In summary, then, this is a complex interaction between sun exposure, vitamin D synthesis, some inherited variation on melanoma susceptibility, and somehow we have to convert that into public health advice. Thank you.
Great. Okay, thank you very much, Julie and David. So we have a few questions. So the first one was about the effect of, was about the effect of neonatal blue light on the risk for melanoma. Is this something which has been taken into account in any study so far? And could it have any effect on the overall risk for melanoma, at least in young people?
I'm not aware of any studies. Are you, David?
No, I was just trying to rack my brains, and I have some vague memory that maybe Tony Swerdlow looked at this some years ago, but I'm not sure if it ever made it through to publication. But interesting hypothesis, and it sounds like it would be an interesting one to follow up.
Yes, there was an early study, wasn't there, from Val Beryl suggesting that fluorescent lights were causal, and Tony Swerdlow did studies subsequently but didn't confirm that association. But I don't know of any. Of course, it'd be quite difficult to do such a study, wouldn't it, because of although increasing relatively small numbers of babies treated, relatively small numbers, it would have to be something like a registry study if we were able to capture
a linkage study,
I think.
Yeah.
Okay, Martha Shell, who was the one who asked this question, actually is writing here that they have data showing that this kind of exposure is actually leading to an increase in the number of nevi. So this may have perhaps some effect on melanocyte proliferation. Another one is how much have we learned about non-sun related elements in melanoma pathogenesis from the non-cutaneous forms of melanoma, or those melanomas who are developing in non-sun exposed areas like rectal melanomas?
I think we don't know. I'm sure David has a view, but all of these sorts of melanomas are quite rare. All of the studies are relatively small, therefore, and I have long argued that what we need to do is a multi-country study looking at etiology. That people have always postulated that acral lentiginous melanomas may be related to trauma, and some have plotted the incidence according to pressure on the foot, but I don't think it's very convincing evidence. These tumours are very biologically different to melanomas in other sites, and as far as I'm concerned, it's very difficult to identify any evidence that's persuasive about etiology. But what do you think, David?
Yeah, there have been some epidemiological studies looking at acral lentiginous melanomas particularly. I know Adele Green's been looking at those, and the idea of trauma has been explored, and there looks like there's some evidence for that, or at least people recall history of trauma. Whether there's a recall bias going on or not, it's hard to say. When you look at the mutation profiles of those mucosal and acral melanomas, though, they are completely different from the cutaneous melanomas, and they have much lower mutation burden, lower than the cutaneous melanomas, and it appears a different constellation of driver mutations as well. I know Nick Hayward from Brisbane published a paper in Nature last year or 2 years ago with the sequencing data of the couple of hundred of these acral and mucosal melanomas, and they really are quite distinctive. So what's causing that in the environment is not completely clear.
I think there's some suggestion that acrals have more translocations and major chromosomal changes, but who knows why that might be.
So we have another couple of questions here. The first one is, are there any genetic susceptibility factors for melanoma that affect the immune system rather than pigmentation?
Well, I've been very disappointed that there haven't been hits in that area. On that GWAS Manhattan plot that I whizzed over, there were blue peaks, and these are of unknown function biologically in terms of melanomas. So it's possible that may be so. We have done lots and lots of work looking at candidate gene approaches, but they're always, more. We can't rely upon such approaches, but we've been looking for immunological genes for years, but we haven't actually found strong evidence of that.
Okay, and also, what is known about the mechanism of photoadaptation in terms of epigenetic mechanism, maybe immune regulation as well?
Well, I am absolutely not a photobiologist. However, broadly speaking, of course, quite a lot of epidermal thickening occurs when you're chronically out in the sun. But I don't know anything at all about the epigenetic control of that. I have an interest in vitamin D signaling, however, because that's where our data took us. And of course, vitamin D is synthesized in the skin. And vitamin D, from a paper we recently published in Cancer Research last year, we published biological evidence or evidence for causality in terms of vitamin D receptor signaling in the control of melanocyte proliferation. So I can't really say anything more than the suspicion, pointing to the suspicion that local production of vitamin D in the skin when you're sun exposed is important. As I said, we've looked at vitamin D receptor signaling, and I have never really understood why melanoma on the trunk has a poorer prognosis than melanoma on the limbs. And I was therefore interested to see that the vitamin D receptor expression in our tumours that had been removed from the trunk was significantly lower than in other sites. So one hypothesis we have is that with chronic safer levels of sun exposure, there is an induction in vitamin D receptor, and that plays a role in inhibition of proliferation of melanocytes. So we were talking about mucosal melanomas before, or acral lentiginous melanomas, and not surprisingly, they therefore have very low levels of vitamin D receptor expression. So we continue to pursue our understanding of that, but that's all I know, I'm afraid.
So first of all, I'd like to very much thank you, Julia and David, for this beautiful presentation and the discussion. I'd like to thank all the participants, and I'll pass it over to Chris for a final word. Chris.
Okay, well, thank you very much, Ellie, and thank you particularly to David and Julia for doing such an outstanding job with our first Sweet and Sour. It's going to be a tough act for anyone to follow, so it was entertaining and educational. I'm just going to close by giving you a glimpse of what we've got coming up in the next couple of sessions. I'm afraid you've got to put up with me for a whole half an hour in 2 weeks' time, where I'm going to be leading off the batting for molecular cuisine and talking about my personal and somewhat variable path of discovering psoriasis. And then 2 weeks after that, on the 19th of August, we'll have the first of our freshly baked sessions, fronted by Cynthia Orvain from Paris and Wim de Klerk from Ghent. We're going to discuss, as the name suggests, very recently published high-impact papers, and I'm looking forward to that one particularly, as to how we're going to be able to understand various in-depth new studies. So thank you for listening. We welcome feedback on what you think about these ESDR Kitchen events, and look forward to seeing you all, and perhaps not even seeing you, but certainly having you participating in 2 weeks' time. Okay, goodbye.